this is where we left off last time, I believe. And I did a lot of narrative, shall we say, last time. And so a lot of these slides have already been explained. So I'll go through a lot of these at what may seem to be a rapid pace. I hope it's not. Should be somewhat familiar to you. So let me cut the sound down a little bit if I can remember how. Testing one, 2. Okay. So we're talking about RNA polymerase and its structure. We're getting ready to transition into how it functions at the domain level within the active site of the enzyme itself. And a way to do that is to go from a subunit discussion, which is what we've begun, to an example where there's only one subunit. In fact, it is all the subunits of the bacterial enzyme are combined into one peptide for a lot of the phage. And this is an example of phage T7 RNA polymerase. And you'll notice it, well, you don't know it yet, but it has the same overall conserved structure that you get when you have 4 or 5 subunits. This big cleft where the DNA goes, and you see the DNA takes almost a 90-degree turn as it hits one surface of this cleft. All of this is very similar to the bacterial model. And notice here that the speed, though, is a lot faster than the bacterial RNA polymerase, about 200 nucleotides per second. That's really sailing along, whereas you remember how fast the bacterial RNA polymerase goes? 40 to 50, which is about the same rate as the ribosome travels as it chases it. So here you see the conserved domains of this single peptide in the phage. If I could back up, that would make it it's just not backing up. We'll have to get manually back up. Oh, I guess it backed too far up. Okay, this is the correct slide. This is one that did not get completely converted, so occasionally I miss these. It should be, the text should be in blue and so forth. But what you see is thumb, fingers, and palm. That's a conserved domain structure of RNA polymerase, including the one that we have. Notice here that sigma, what is normally sigma in the bacterial enzyme, is still part of the same single peptide or protein. And let's keep going here. You'll notice the sizes of the transcription bubble and the RNA DNA hybrid are all approximately the same. It covers a space pretty small, from -10 to +10, so it makes a fairly small footprint. Note that bases of the template strand are flipped out. You know, this base flipping mechanism we were talking about. This strand here is the template strand, and they are rotated so that they have a better access to the incoming ribonucleotides. So base flipping is widely used in lots of enzymatic processes. Put most of your 95% of your emphasis on the bacterial enzyme, even though there are several slides on the phage. I don't know whether there'll be questions on the phage or not, but remember the bacterial thing, structure. And here you see it. You have this groove that the DNA enters. Here you see it here. You should, from a picture like this, you should be able to identify all the components. Variations of this picture show up on occasion. Notice that the DNA here is red and yellow, and pay attention to where the RNA is. Can you see the RNA? There. It's growing from the active center. It's just beginning to grow. It's going to exit right underneath here, the exit pore. It's going to be underneath and it'll come out right back there. Notice the non-coding strand is just displaced, looped up, totally out of the way. It serves no function during transcription. The template strand would be the one in red here. And you see the nascent transcript. What does nascent mean? In the process of being formed. So a nascent peptide would be one still stuck to a ribosome in the process of being made. A nascent transcript is the RNA that's still in the process of being synthesized, still attached to the enzyme. Okay, keep going here. Can you label the parts? You can't from this tell what's beta and beta prime. I'm not going to, I mean you actually could, but I'm not going to ask you to know anything except non-coding and coding strand and transcript if you had to label parts of this. This is same kind of representation, but here you can see the groove a little bit better. It looks like some kind of yapping dog. This is the main channel. The active site is right back in here. That's where the magnesium is. The beta prime is on top. The beta is down below. And now we've got some other things. Omega is a subunit not talked about much. It's used for assembly and sort of holds it together. And then you have these alpha subunits, and you have two of these. They're colored yellow and green here. These at the end, we'll see more about those in a minute. Here you see another representation. It looks like some Tyrannosaurus Rex skeleton at the National Museum. This is a favorite one. I have so many favorite figures that I like to put on exams. I like to share them with you. Here you see, oh no, this thing has lots of tricks. RNA polymerase passes through several configurations. What you'll see is that RNA polymerase, it has the jaws, and the jaws have essentially two states, open, closed. When it's open, it doesn't have as high affinity to DNA as when it's closed, naturally. When it binds sigma, it pops it open a little bit, and that allows it to hop and search for a promoter. There's another aspect of sigma in that at first, before DNA is present, a part of sigma actually occludes or lies within the groove that DNA is supposed to occupy. This end of sigma is called region 1.1, it's at the N-terminus, is negatively charged, just like DNA is negatively charged. It occupies a similar pocket. When this binds to DNA, that end domain of sigma, and sigma is shown here as this red cluster here, and you see the negative charges around the extreme end of the sigma subunit. It rotates upward. See, it went from down here to there, making room for the DNA to enter. So the first thing you see is the DNA makes contact with the sigma, but it's not in the main channel, the DNA channel at first, because the domain 1.1 of sigma is in the channel, and then it flips over and enters the channel, and this domain 1.1 swings out of the way, and notice how it is bent here. The template strand remains attached to the wall, and the non-template strand gets displaced. Here you see it here also. And so when sigma leaves, because this is an open promoter complex now, sigma's getting ready to leave, the jaws clamp down a little more. So this would be the configuration when the core is bound to non-promoter DNA, there's no sigma, it's going to be held pretty tightly. so it doesn't hop very frequently. When sigma binds it, it opens up, now it can hop. When it finds a promoter, initially it is still open, but then in the process of melting, it begins to close, and when initiation is finished and sigma leaves, it leaves the polymerase clamped back down again, but as it's elongating, which is perfect, which means it's not going to fall off of the DNA very readily. It increases processivity because of that. You'll notice it has an initially large footprint, which then contracts to the smaller configuration as it elongates from -30 to +20. Of course, this is before it actually leaves the promoter, because it's +20 -30, it is sitting at the promoter, but it's making this little abortive transcript. This is undergoing the cycling. This is what yeast looks like. You notice it looks very similar. Of course, this is a very simplified model, but so you can talk about yeast and bacteria and even phage. It's the same engine, the same machine that transcribes RNA. Once you invent it, you don't have to keep reinventing it. You just kind of change the configuration of subunits, but you have the same structural domains. And so you'll need to know these structural and functional domains of the enzyme itself around the active site. You'll need to know the wall, the bridge, the rudder. Notice the nucleotides come in from the bottom. There are 2 pores, one for incoming ribonucleotides, and then out the other side, it'll be the place, the transcript here is in yellow, it will be exiting this pore here. So let's get a little more turn. Notice that this slide is emphasizing the sharp turn it's made. The DNA is kind of bent, and this facilitates its melting, actually, this bend. It's bent by being slammed against this wall domain of the active site. Of course, the magnesium is going to be right down here where the nucleotides are being added, right at the 3' end. Know the functions of bridge, rudder, wall, and clamp. Can you label the domains and pores? DNA is forced to make a turn at the active site by a wall. This helps flip the RNA away from template strands, so it's part of tearing the the transcript away from template, something you would not see happening in a DNA polymerase. You wouldn't have that strong bend. It also facilitates the melting, you'll see down here. Yep, I get the hang of this pretty soon. The melting down here, and the flipping, all of this bend does lots of stuff. The rudder helps separate the new transcript, the nascent transcript, from the template. It sort of gets in the way so that the rudder, that the transcript gets dislodged off of the template by running into this rudder structure. Double-stranded. DNA is very flexible, but if you cut it into smaller and smaller pieces, it becomes more and more rigid. So it's fairly rigid if you have only 200 base pairs. But if you have a KB, you can twirl it like a cowboy in a rope, but the shorter you get it, the less flexible it becomes. So all of these events here span a region that's a lot less than 200 nucleotides. So this is a big deal to put that kind of turn into DNA over such a short space. How does the enzyme bind to promoter without the DNA hitting the wall? A large change in conformation must take place. The clamp is initially out of position and repositioned to keep the DNA in the active site after it is melted. So this is a very dynamic process, the binding, then the melt, and then abortive cycling, and then initiation. We'll get into the specifics in just a moment. This is the big challenge for the enzyme. You need to properly align everything. Everything is position, location, location, location with most enzymes. You get the right position, and then the catalysis or synthesis can't help but happen. It just happens simply by probability based on positioning. So how do you keep this very precise alignment of the incoming ribonucleotide and the 3' hydroxyl of the growing RNA transcript? How do you keep that alignment intact if every time you incorporate something, you have to pick up, release from the DNA, take one step forward, and now clamp back on? That's what the enzyme essentially does. And so while it's opened up, taking this one nucleotide step before it clamps again, it'd be nice to have like a third arm or something, right, that would hold on while you're taking one step. That's what the enzyme has. That is called the bridge. So this structure here, this is the bridge. It's going to work like this. So the enzyme is going this way. My nose is the 3' end of the transcript. It's going to grow. And right in front of me is the space for the incoming ribonucleotide. It's going to come in here, attach. And then I have to move one base forward, and then attaches again. So the bridge is like this. It's in contact. It's right here. There's a hole here, and we're waiting for the ribonucleotide to come. So it comes up here, attaches, and then I take one step forward. I'm sorry. It's like this. I get it mixed up. It attaches, and then I take one step forward, and then I'm in the new configuration again. So the new nucleotide comes, and then the enzyme walks forward, but the bridge remains in contact. So much fun to do that. I only get to do it once a year. In the spring, the bridge, it remains in contact with the 3' end and allows the polymerase to loosen its multiple contacts and then reform them after every nucleotide. And then in the book, there's probably 2 pages trying to illustrate what I just did. See that silly thing? They could have just had a guy walking across the page and it would have done the same thing. Snaps back, all that, we know that. The dilemma of how to do it goes on and on. We've already been there. The bridge, the bridge, the bridge, really? Okay. Which domain of RNA polymerase prevents the RNA-DNA hybrid from getting too long? Uh-oh, I just said it in passing. It's the rudder, isn't it, that tears the new transcript away from its mama template. Yep, must be. This is the hand puppet. Almost looks like fingers, doesn't it? This is the core, and this is yeast. You see there are 2 essential positions, open and closed. When there's sigma, it's open. When there's no sigma, or when it's in the highly progressive elongation state, it is closed. So you see that these 2 polymerases are in 2 different configurations. You know, if you took the bottom one here and fused them together to make a mask, you could scare the daylights out of your cat, couldn't you? Yeah, whoa. Sneak up with that thing. Kitty, 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 kitty. Ah. Okay, I digress. This is like the modern art version of the RNA polymerase. I will not have this on an exam, but what you'll see is this is the eukaryotic version. Yeast looks pretty much like ours. You've got the beta prime, the beta, and look at all these other subunits that surround it in the eukaryote. They constitute, if you looked at it, its surface topology, you would see it's the same enzyme. Here you see the yeast with the subunits. Notice it looks very similar to the bacteria, even though it has lots of subunits. And here you see the DNA. It's being pretty much surrounded by this DNA channel. What is this red thing here? Is that the tongue? That's the magnesium ion at the active site. Here you can see another view looking now with the DNA laying pointed at you in the jaws. Here you see the subunit structure of bacterial RNA polymerase, which is something that you pretty much know now, so let's look at it. The beta prime, of course beta prime is the largest, and you have 2 alphas. You see the 2 alphas, and then 1 omega here to form a core, 13,000 molecules per cell. Almost all are bound either to promoters in the process of elongating or trying to figure, or just sitting on non-promoter DNA. Notice how large it is. It's roughly 500, 460 or so kilodaltons, pretty large. Then we've added the last subunit, sigma, and this is the brains. This is the map disk that you put in your Garmin that tells the polymerase where all the promoters are, that it's actually found a promoter, sigma. And sigma, of course, is only needed when you're searching for a promoter and when you're trying to melt the strands of a promoter. Then you don't need it anymore. Here you see another view, and this, I've seen this on exams before. That's that magnesium ion, but this is flipped around the other way, so the DNA channel must be on this side, the beta prime, the beta. These 2 things, subunits here, the alphas now are a little bit more clearly seen in their true nature. Alpha 1 and alpha 2, it's the same, 2 copies of exactly the same protein, and you see this flexible linker and a C-terminal domain, CTD. So the N-terminal domains attached to the core, and the C-terminal domain, and there are 2 of them, these things will kind of bounce around like tin cans on strings behind the wedding car of polymerase, and their job is to enhance or regulate promoter activity. They're not catalytic in any way. They have affinity for a special element that's upstream of the promoter proper. It's called the UP element, up. They actually bind it if there's one present, but quite often there's not one present. There'll be a protein instead that is quite willing to bind them in its place, because you can regulate a protein whether it's going to be there or not. So now you have a way to regulate the activity of a gene, because you can regulate the breakdown, stability, and synthesis of a regulatory protein that interacts with the C-terminal domains. Does our RNA polymerase have a C-terminal domain? Was this invented by bacteria and it just died out, or is this idea, did it gain some traction and get elaborated in us? We have a quite extensive CTD, and it does not bind DNA anymore. It exclusively binds a whole host of processing complexes. And you see it being dragged along behind only one of the RNA polymerases that eukaryotes have. They have a minimum of 3 RNA polymerases. Bacteria just have 1. Archaea just has 1. So we started out with 1, but we've become specialized, so we now have 1 devoted to the ribosomal cistrons, that's RNA polymerase 1. Polymerase 2 is devoted to protein encoding genes. That's the one that has this long CTD that strings out behind it that has binding sites for RNA processing, capping, poly-A addition, lots of stuff. So the idea is quite ancient that you can put strings on the end of your polymerase for regulation. We only keep that motif of regulation for Paul II, but we really exploit it. The alpha subunit has C-terminal domains. They interact both with DNA and for regulatory proteins. So let me see here. Which subunits from the catalytic center? Beta prime beta. Which subunits required for specificity? The alpha. What are the 3 roles of the alpha subunit? Uh-oh, 3 roles? Ooh. 1 to recognize a DNA element called the UP. 1 to interact with regulatory proteins. 1 to perhaps stability. I don't know the 3rd one. I didn't see it in the previous slides either. Assembly. Does it say up here? Recognition. Assembly. Assembly. These are 2 alphas. So the omega here, right here, it didn't say what it's for. This has an assembly role also. So either the slide got it wrong or I forgot something. Or both, who knows. You won't need to know, but 2, there's not a list of what are the 3 activities of alpha. Here you see all the contacts that are made, multiple contacts that are made with both well, primarily with the DNA, but also with the RNA as it's growing, beta prime and beta. And a lot of these contacts turn out to be on the same helical face of the DNA, which we'll see in a minute. And which makes sense. The enzyme sort of grabs around the DNA in the channel. This the DNA is here. We're looking at it on the end. And so the bottom helical half will be in contact with DNA instead of all the way wrapped around it. Okay, so this is a close-up look at the active site. And this illustrates the connection between the scrunch and the size of the abortive transcript. So what am I talking about? You remember in abortive cycling, you go 1, 2, 3, 4, 5, 6, 7, 8, 9, and then if you can't cram or scrunch any more DNA into the interior of the polymerase, you have to let go of the tension, and this little transcript floats off. So the size of this little abortive transcript that floats away should be the same size as the number of nucleotides of template you can cram or scrunch into the enzyme without having to move. Because all of this abortive cycling happens with the polymerase stuck firmly to the promoter. So technically, you're making transcript, but it's still called the initiation phase of transcription because the enzyme has not begun to move down the template and move away from the promoter. So it's stuck at the promoter. Why is it stuck at the promoter? Seems crazy. It's already started transcription. Why does it go through this little song and dance? It's stuck there because it's got this maniac subunit called sigma that just like a Tasmanian devil, it goes promoter, it just chomps down, it can't let loose. You've got to convince sigma to let loose, like a snapping turtle. how does the enzyme convince sigma that it's okay to let go? So that the enzyme now can go 10, 11, 12, 13, 14, 15, and keep going. How does it do that? It does it by, I guess, poking sigma in the eye. Piggly wiggly. What's it? Cat hips, fish lips, bird bath pie, piggly wiggly. Everybody goes poke you in the eye. The nascent transcript. That's how you pass a meme on to the next generation. The nascent transcript tries to leave the RNA exit pore, but it can't because sigma has its head stuck in there looking at it. Like one of those moray eels, it's got his head stuck in the exit pore, and that nascent transcript just keeps bumping it in the head, and eventually it'll push it out of the exit pore. When it pushes sigma out of the exit pore and begins to exit itself, that changes the configuration of sigma. It goes, whoa, and lets go. That letting go, it enables the polymerase now to let go of the promoter. That's the only reason it's stuck at the promoter. The leaving of the nascent transcript after many cycles of abortive initiation changes the configuration of sigma so that it drastically loses affinity for both the DNA of promoter and for the RNA polymerase itself. They used to say that sigma just completely, fell back on the floor, completely disassociated from the core. But now we know that it's not an all or nothing thing. It just loses the ability to make the core bind tightly to the promoter. It sort of diffuses off gradually, kind of slowly leaves as the enzyme goes along. It'll just fall off because it has very low affinity now, because it's, had its clock reset or something, as they say. So let's look how this diagram works here. So can you recognize some parts? This is the template strand here, and here you can see the RNA chain. No, this is the template strand here, and right up here is where you can see that's the 3' end, and here's the next nucleotide being added. So it's going to be a G added to the C. The CG right here has already been formed. So this is the new nucleotide coming in. This is the growing chain. So the 5' end you can't see is up there someplace. And so as it goes 1, 2, 3, 4, 5, 6, 7, 8, the DNA gets kind of scrunched around into this area. Into that so-called hydrophobic pocket. Hydrophobic pocket, what's the significance of hydrophobic pocket? The bases themselves, the interior of DNA is hydrophobic, excludes water. So if you're going to bind the single-stranded bases in this, you'll need a little hydrophobic pocket to bind it. So that's where the scrunching happens. You can see illustrated here, a flipping occurs. The next base flips over so it can be read. Here you see it flipped according to the diagram. Can't quite see it until I squint my eyes. Okay. So understand what DNA scrunching is and what its relationship is to the size of the abortive transcript. They're the same, 8 or 9. 2 to 9. Yeah, 2 to 9 is what it is, not 8 or 9. What do you need to know here? What I usually ask here is, at this you'll see the stages in going all the way from holoenzyme bound to non-specific DNA all the way down to RNA synthesis, promoter clearance. You'll see that the holoenzyme, this is with sigma, it starts as a closed binary complex. Why binary? It has 2 components, the enzyme, which is protein, and DNA, 2 components. When it gets down here, it'll be called ternary because there's an RNA transcript being formed. And this step right here, once the open binary complex is formed, that's when the strands are open, it's considered irreversible. Then you get the ternary, which means you're making a little abortive transcript, and one of those elongates for the real message. Okay. Sometimes I have something like this. I'll give you that image and you'll go, I don't know, it's a blue blur. Nope, that's core with no sigma because it's closed versus a core with sigma because it's open. So I'm testing whether you realize that the addition of sigma opens it up so it can now scan for a promoter. You remove sigma and it's useful in two instances when you have it closed. One is when you're just resting on non-promoter DNA. And the second is when you're actually elongating and you want the enzyme to remain stuck to the template. This is illustrating the fact that sigma, drawn here in sort of pink, its domain 1.1 actually partially occludes the channel. Before the DNA gets there, here's a more accurate representation. Sigma domain 1.1 is in sort of pink, and you see it before the DNA enters. There's not quite enough room for the DNA to enter too, and this sigma moves out of the way, and now the DNA can fully seat itself in the DNA channel. Here you see that same concept illustrated again. This is sigma in red. You see how it's a very convoluted protein, winds and weaves all around. And this is domain 1.1 of sigma. And that domain rotates slightly out of position here. You can't see it very well, but that's as the DNA seats itself, partially hidden there by the lip, the sigma rotates out a little bit. In addition to sigma moving, the jaw itself closes down. The jaw, here the end of the jaw is closed right there. That's the end of transcription part 1. But look, we can go to part 2. Any questions on part 1?
So what was the enzyme that needs to be kicked off for abortive cycling to stop?
Sigma has to be dislodged before the core polymerase can leave the promoter and enter into the elongation phase. In fact, that's a favorite question of mine. What event triggers the transition from initiation to elongation? That triggering event is the dislodging of sigma by the 5' end of the nascent transcript. As the transcript tries to exit the RNA exit pore, it dislodges a domain 3.2 of sigma, pushing it back out of that exit pore, and that causes sigma to lose affinity both for the promoter and for the core enzyme, allowing the promoter now to leave the promoter and begin to elongate.
But it's also required to bind to put it in the open conformation, right?
You have to bind it first before the core can recognize even what a promoter looks like, yeah. And to open it up so it can hop and search. You don't need as much sigma, you need about a ratio of 1 to 3, because it's only there 25% of the time engaged. If you look at that, remember that pie chart? The top half is core bound to non-promoter DNA, 50% of it. And then down at the bottom of the pie chart, you've got two slices, 25%, 25%. The first 25% over here on the left is sigma bound when it's looking for a promoter or melting the promoter. And sigma is not a part of any of the rest of it. And the other 25% over here is the percentage of core that's actually elongating at any one time. Okay, let's go to the next one here. Let's look at the actual structure of the core promoter. And we're going to talk about sigma and how sigma makes contact with the core. If you were designing a promoter, a place to start transcription and establish the polarity of synthesis, they're the two things you have to do, right? Not only find where to start, but which way to head, right? You could head the wrong way. So there has to be information there as to which way and where. You have to have a minimum of 2 contact points to make that go smoothly. An A and a B. A comes before B. And so promoters in their simplest have 2 points of contact. They have a -10 site and a -35 site. And they are unique. And so that establishes direction. The -35 is, by how you count, is always going to be further upstream than -10, right? It points the direction, points the polymerase in the correct direction and identifies where it should start. And so here you see the promoter. Along the bottom, promoter is the DNA, and it covers about 75 bases sometimes, but it's not all conserved. Within it you have little blocks of conservation, and those little blocks are points where the enzyme makes contact and specifically recognizes it as promoter. And so let's look at these little blocks that represent core promoter elements. The -10, if you start, +1, right? +1 is the first nucleotide of the transcript. There's no 0. +1 is the first, is the end of the transcript. -1 is the first of the promoter. So +1, right here, well, the two main ones that you learn in previous courses might be the -10 and the -35. We'll learn that there are many sigmas, and each has a slightly different sequence and a different place they bind. But for the most predominant, by far, sigma has a molecular mass of 70 kilodaltons. So it's called sigma 70. It has a site at -10 and a site at -35. You see that they are different sequences. The first one says tata. One of the most highly conserved bases of the tata is actually the last T, strangely enough. And here you see the subunits as they lie along this promoter. So you have this region here. Let me get the next. Okay, right there. The alpha subunits, the alpha CTD, remember the 2 alpha subunits, they bind right next to each other at an AT-rich stretch that's called the UP element. Many genes, many promoters do not have a site for the CTD to bind directly to DNA, but they will put a regulatory protein here that will in turn bind these. So these will be bumped up and there'll be a protein here, and the CTD will be making contact with, for example, CAP. The CAP is the cyclic AMP binding protein. So CTD binds here directly when it's a UP element, or it will bind to a regulatory protein. And all the other contacts are made with the various domains of sigma. And you will be responsible for all of these domains. So before we leave the promoter, your up element, the minus 35, the minus 10, and there are a couple of other regions that are important. Some promoters have an additional recognition element called the discriminator that makes contact with sigma, and it's between the minus 10 and the plus 1. It's kind of an obscure element, but it's very real. Makes contact with region 1.2. The minus 10 and minus 35 are the predominant interactions. Notice here that contact is being made between one of the alpha subunits and sigma itself. That's very important. When the CTD is bound, either bound to cap or bound directly to a UP element, it helps to stabilize sigma. Why would it need stabilization? Because a lot of times the minus 35 is very hard to recognize. In some cases, it's not even present. In fact, you see this perfect promoter? Almost no genes have a perfect promoter, right? Why would you want a perfect promoter? Because you wouldn't be able to control it. It'd be a wild child. It'd be wide open all day and night. It could not be regulated. It's got everything it needs. It's perfect. I'm perfect. You can't induce me. I'm not shutting down. You can't shut me up. And so promoters have to be broken all the time. So they need a little help. They need an accessory protein so they can be regulated. Positive and negative transcription factors serve that role. And this is one of the ways alpha, if it's able to bind a UP element, sometimes there'll be something on the UP element so it can't bind, and then this kind of interaction can't occur, and now this poor minus 35 has a hard time holding sigma. So you build in a mistake, and quite often the mistake is at minus 35, so that you need to have this further stabilizing interaction between the CTD of alpha and sigma, or between cap and the domain 4.2 of sigma. Very deliberate strategy employed by all organisms. Build in mistakes so you can regulate. So here, this is sigma, and this is the alpha subunit. I was telling you there are lots of sigmas. Sigma 70 is by far the most abundant and works at the vast majority of the promoters. It's so-called housekeeping sigma or sigma 70. But you notice there's a whole host of others that have been specialized to their own little collection of promoters. What does that mean? It means those promoters don't have a minus 10 that looks like the one that sigma 70 binds. In fact, you'll see here, well, you can't see it on this one, this next slide. What you'll see is they don't look anything at all like Tata. That's so sigma 70 leaves them alone so they can regulate. But before we go into the sequence, we'll just look at the functions here. Sigma 70, let me point out the ones you'll need to know. Sigma 70, sigma S. The S stands for stationary phase. Why would you need a special sigma for stationary phase of a culture? Well, in stationary phase, you are starving, right? You're a Marvin. You want everything to be slowed way down because there's not enough food to go around. You don't want to have really active metabolism or transcription or gene expression. So you've got a sigma here that doesn't work properly. Deliberately. So it tones everything down. And let me go back here. You notice it says contact between sigma and the alpha CTD in sigma S is negatively charged, so alpha can't bind. Sigma, the stationary phase sigma, cannot bind to the alpha, so it cannot be stabilized at the lousy promoters that don't have a very good minus 35. So that's an easy way to regulate. And that sigma S looks identical to sigma 70. Even the sites where it binds is the same. But all the rest go their own way. But that's the mistake or the flaw in sigma S for stationary. Okay, we got that out of the way. Heat shock. If you suddenly elevate the temperature of anything on Earth, it evokes a universal response. You usually get very robust gene expression to just a small subset of your genome, and these genes quickly turn out vast quantities of proteins that assist in folding and refolding, and proteins that keep your proteins from denaturing into insoluble aggregates. In fact, some of the proteins that are produced form surfaces that other proteins can aggregate against instead of aggregating with themselves. They're called chaperones. The small heat shock proteins do that. So you have these binding surfaces. So an enzyme may denature and its hydrophobic insides will be all exposed. Normally they would, do a belly bump and aggregate with each other irreversibly. But if there are lots of small heat shock proteins around, they bump into these big beach balls and they bind them. And now they can be reconstructed and refolded off of the beach balls and have a happy life after the heat shock has gone away. So now sometimes we call this a protein unfolding response. Heat does it. How do you evoke the heat shock response? How would you do it yourself? If you cut the water on in the bathtub and got it really hot and ease down, as you made contact, you'd be heat shocking. All you need to do is elevate the temperature 10 degrees centigrade. A sudden elevation in temperature of 10 degrees centigrade will universally activate the heat shock response. In fact, that is so easy to do, that's what my laboratory in the early 80s decided to take on as a transcriptional system in plants to see which promoters and which proteins are responsible for making that happen in plants. Okay, enough about me. Sigma 32, heat shock. What's another one? Sigma 54, where are you? Nitrogen assimilation. If you had starved for nitrogen, you can kick in some pathways that make this go more efficiently. And what's the last one? They're the only ones. 54, 32, S, and 70. They're the ones you'll need to know. And look over here. These are the actual promoter sequences. Sigma 70. There's the TATA at minus 10, and here's the TGGACA, the minus 35. So both the sequence and the distance between them is a part of the configuration that's essential. If you wanted to make a promoter or gene less active, just start inserting bases between the two control elements and you will decrease its efficiency. Or if there are too many there, remove some. If you get it within the 16 or 18 base space between minus 35 and minus 10, it will be optimum. Of course, any mutation toward the consensus makes it more perfect. And of course, less easy to regulate. So look at S, sigma S for stationary. It's exactly the same sequence with the same spacing and everything. So what is the mistake? S cannot make contact with the alpha subunit. So it can't be stabilized and assisted in binding by contact with alpha. Stationary phase. And look at some of these others. Here's one. That doesn't look like much of a tau-tau, does it? And it's only separated by 6 base pairs from the so-called minus 35. Obviously, that's not at minus 35 anymore. So the main theme here is that there are 2 elements, 2 main elements. That establishes specificity with respect to which sigma, and it determines direction of transcription. 2 elements. Here you see the number of promoters controlled, 1,000. For the heat shock one, which is 32, only 30. For nitrogen starvation, only 5 promoters use that sigma. Some organisms some bacteria undergo sporulation. They form spores under very environmentally stressful conditions like, what's it called? Bacillus subtilis. Bacillus has about 19 sigmas or so, and it's a cascade of them being activated. So that's called development if you're a microbiologist. Of course, development if you're a human geneticist means the changes in the organism from a zygote to a fully grown human being. But forming a spore is about the only trick a bacteria can do. That's called development. I like to look down my nose at microbiologists, being a plant guy. I'm looked down enough on as it is by the human geneticist. I gotta look down on something, right? Slime on a plate, you call that an organism? Don't tell Monica I've said any of this, right? Okay. So let's look at even more detail of sigma. Uh-oh, spring break just started. Okay, so see that little thing there? That little loop there is what pokes in the RNA exit pore. And this is what the 5' end of the nascent transcript bumps up against. That is domain 3.2. That's important to know. That little hairpin there, that is what is poking into the RNA exit pore and interacts with the 5' end of that growing transcript. It looks like an accident, doesn't it? We do the same thing. Our polymerase works the same way. We don't have anything called sigma, but we have something that has very little resemblance. But there are certain domains that look like this and do the same thing. It's called TF2B. That's for module 4, so just forget that. Okay, here you see domain 4.2. Notice how this is kind of flipped around from the normal Gutenberg orientation. This is now going, I guess this is how it comes off. So anyway, the end, usually you see these, well, a lot of times you see it this way. A lot of times you see sigma written the other way, but maybe that's just me. This is the C terminus, and this is the N terminus. This is 1.1. This is, you remember what is protruding into the channel of the polymerase. 4.2 has a helix turn helix. How many have heard that term, helix turn helix? What does it do? Helix turn helix. It's almost a unit. Well, you got a suggestion? No, you don't want to say, but you know. It's a universal motif, or fold as they say, that recognizes and binds DNA. DNA in bacteria is recognized by a helix turn helix. One of the helices sort of sits down in the major groove, and the other 2 helices stabilize it with respect to the phosphodiester backbone of the 2 parallel strands running. So you get this big dip, and the one helix sits down, and the other 2 help configure it with respect to that major groove. That is the predominant way bacteria interact and recognize DNA. They bind to the double-stranded DNA. They don't open it up and read the bases like we have to do. It can just kind of, by just touch, it can, oh yeah, on the outside they can say, oh, that's got to be an AT pair or a GC pair. Took them 1 billion years to learn how to do that, but they're pretty good at it. They don't even have to open it up. Helix turn helix in domain 4.2. And if you look right here in this region here, this is a special region. This region is involved in recognizing the -10. And it's really sort of one long helix or 2 helices. It's a bent helix, but it's definitely not a helix turn helix, because a helix turn helix is really 3 helices. It's a triple helical cluster reinforced by beta strands. The name is misleading. It's really helix turn helix. So these are some of my questions. This is a very unusual domain for recognizing DNA, just one long helix followed by another long helix at a 90-degree angle. This has to melt DNA. Normal DNA binding proteins that control transcription, they don't need to melt the DNA. They're not trying to transcribe anything, they're just trying to bind. But this has adapted so it recognizes and then can melt in between the strands, because this needs to open up the promoter. And the melting is done with domain 2.3. We'll see more about that in a minute. Okay, let's keep going here. Helix turn helix, region 4.2, that recognizes -35. The Tata does not involve helix turn helix. It doesn't have a name other than the sigma interaction domain, but it's not a helix turn helix. What does this region contact? Well, that contact, I thought it just poked through the hole. It makes contact with the 5' end of the nascent transcript. Be familiar with these regions. Discriminator 2,3, 2,4, 3,2, and 4,2. We'll see more about that in a minute. Region 2 makes contact with the core, makes contact with beta prime. This region here is very conserved because it does bind sigma to the core. Notice this is that area that makes contact with -10, 2.3 and 2.4. 2.4 actually recognizes the TAT, and the 2.3 is involved in melting in. And then this helix here, 2.2, recognizes the core. Region 2 is in general the most conserved portion of sigma. (2.3) melting, it has aromatic 2-tryptophanes that melt in between the strands. So this is recognition. 2.4 specifically interacts and recognizes the -10. In fact, it can recognize the -10 before it's melted open, as you might expect. And as it melts it open, it begins to further recognize the fact that it's on the inside too. It has recognition surfaces for the closed promoter, and as it begins to melt, it recognizes the open promoter too. Alpha helix domain, this is, you see the recognition happening, the first part of the Tata sequence. And notice TAT is about as many bases as you can cram on one helical surface of DNA. It takes about 10 to go around. About half of it would be 5. So one helical surface. Most DNA binding events only recognize one helical surface of DNA. If you have a long consensus sequence for something to bind, you're going to have multiple proteins. It's going to bind here, and then one's going to bind here, and then one's going to bind here, because you can only make contact with one face at a time unless you design multiple DNA interaction modules, like a whole bunch of helix turn helix all linked together. That doesn't happen with helix turn helix, but it happens with things called zinc fingers that we'll learn about a little later. One surface of the DNA. Functional domains of sigma. 4.2 minus 35, 2.3 minus 10, the melt, 2.4 recognition of the tata, 1.1 it goes into the channel. We'll learn one other thing that it does. When it's not bound to core, sigma folds back on itself, and domain 1.1 interacts way back here at 4.2, making this thing so it cannot bind to DNA unless it is pre-bound to core. Because sigma collapses on itself, interacts 4.2 and 1.1, it folds up like a jackknife. When it binds core, it opens up, and now core plus sigma together in partnership go in search of a promoter. 2.4, 2.3 melting, 1.1, 3.2, 5' end of the transcript, 4.2, contacts the minus 35, and it makes contact with the regulatory proteins, one of which would be the alpha domain, the carboxy terminal domain of the alpha subunit, and cap and other regulatory proteins. All kind of nudge 4.2 to make sure 4.2 remains stuck, even though it's a lousy minus 35. A helix turn helix inserts into the major groove. What you'll see is most proteins, almost without exception, recognize DNA through the major groove. There are a few exceptions. They say 1, 2, 3. 3 is all I can think of. In all of biology, and lucky for you, they are incorporated in the PowerPoints, and you're going to know them. There are 2 in fact, in prokaryotes, and we're going to learn about these in a minute, sometime at least. In the alpha subunit, the CTD, it's going to be binding minor groove, but we'll get to that in a minute. So you should know this. You almost know it now because I've said it so many times. Most conserved makes contact with the core. Here you see it again. Mutations affect promoter strength. If you mutate the actual basis of the consensus at the various regions, discriminator, the minus 10 to minus 35, you're going to reduce the activity. In addition, if you alter the spacing between the two, you reduce the activity. Any mutation that makes it look more like the consensus will increase the activity. It's very simple. Now you know how to mutate a promoter for less or more activity. Mutations away from consensus decrease the rate of the closed promoter complex. That's sometimes a wrinkle I put on exam. That would be the initial recognition, right, before the melting has happened. And that's kind of intuitive because minus 35 doesn't involve any melting. The melting occurs, it initiates right here and extends all the way down into plus 1 and past region. So mutations that affect the minus 10 could affect the transition from close to open. Mutations at minus 35 would affect the initial recognition of the closed promoter before you've melted the DNA open. Of course, why do we talk about this? Because the vast majority of all gene regulation, whether you're talking about a bacteria or a eukaryote, happens with this kind of stuff here. At the stage of initiation, the affinity that the polymerase has for the DNA. Right at the beginning, the start of initiation, where the polymerase is trying to recognize where a promoter is. And that general theme occurs in eukaryotes and bacteria. And, oh, I love this picture. You see that right there, that dark hole? That's where that moray eel is sticking its head out. That's where domain 3.2 is poking inside to the active core of the enzyme. In fact, that yellow coil there is domain 3.2 of sigma. The actual little bend part is right here, and you see some space-filling models, and there's another helix over here. This is what's protruding into the interior of the enzyme from sigma, domain 3.2, and notice how intimate the contact is with the 5' end of this gold transcript. This gold transcript, of course, that's RNA. You should be able to label this diagram. This growing transcript, this is where the new nucleotides are being added right there to the end of the gold strand. That's the RNA. They're being added, and this thing is growing, and it's going to want to bend right up here and go out, but it can't because it's bumping up against sigma, this yellow molecule here. What's this, 1, 2, 3, 4, 5, 6, 7, 8, 9? This is what keeps that abortive transcript so small. That's the pore. Sigma is in contact. I need to change the color on that font, don't I, to white. Release of sigma is thought to result from its displacement from the nascent RNA. Whoever wins this little pushing contest here determines whether it's going to be an abortive transcript or whether sigma is going to get pushed out, and now the polymerase can leave the promoter and engage in elongation. This is the trigger for the transition between initiation and elongation. That kind of language, that phraseology is sometimes what you'll see on exam. So, since you guys are going to be medical doctors and all, and probably not going to be combat botanists, I have to add a little antibiotic stuff in here. Here's an antibiotic, rifampicin. It, in turn, it turns out, interferes with this process of the domain 3.2 interacting with the nascent transcript. In fact, it is a mimic domain 3.2. It sits there and interacts itself with the 5' end of the transcript, blocking the exit from the polymerase. So the transcript is coated with this big piece of bubble gum here, and it can't get through the exit pore. It's an antibiotic. It doesn't let go. It binds very tightly. That's how transcription is stopped by rifampicin. And this is what I was talking about, how sigma, when it's not in contact with the core enzyme, it is unable to recognize DNA on its own. So that's a different strategy from what cats and dogs and monkeys and antelopes use, right? And your little baby sister and your mom and dad. They don't do it that way. Our RNA polymerases, they don't know anything. They're like this. I'm sorry, I got ahead of myself. Sigma 54 is, let me back up. Bacteria The polymerase itself, when it has sigma, is able on its own to find a promoter, because sigma is the brains. In us, we have a holoenzyme, 12 subunits. That big, lumbering, like a bag lady, just with all the stuff, all those subunits just cluttered around and dangling from it, doesn't know what a promoter looks like. Our polymerases don't know anything about what a promoter looks like. Eukaryotic polymerases need to have some other proteins go out ahead of time and figure out where the promoter is. And once they prepare the nice seat and the nice big comfy chair and get the TV turned on and the popcorn going, now the polymerase is led by the hand and directed to the promoter. The polymerase doesn't want to fool with, oh, what does a promoter look like? That's in us. In eukaryotes, there are proteins that scout the genome ahead of time, look for promoters, because it's a pretty hard job in us, because most of our DNA is all wrapped up in histones. It's not a simple matter of just scanning the genome and hopping like a little bunny. It's a major thing. How do you find a promoter in a eukaryote when, if it's not being actively transcribed, it's all wrapped up in histones? It takes a while. It takes some planning and some complicated mechanisms. Bacteria, it's not that complicated. So sigma, though, just can't go on its own and find promoters, because if it did, that would sort of inhibit a holoenzyme from binding to that same promoter, wouldn't it? So you really don't want sigma to go out on its own and find a promoter unless it is bound to core. Otherwise, it acts as a competitive inhibitor. So this is how it solves that problem. This is the domain 1.1. It folds back on itself and makes contact with 4.2. Only when it's in contact with the core is it able to open up, and now its recognition domains, 2.4 and 4.2, are able to recognize a promoter. If you delete domain 1.1, you can get free sigma to bind a promoter in vitro. And this looks like, oh, this is showing that domain 1.1, we've talked about that before, how it's initially occupying the channel and then it rotates away. Which subunit of the polymerase holoenzyme partially blocks the entry of DNA into the channel? Sigma's there twice. Sigma region 1.1, not 3.2. How does the holoenzyme transition from abortive cycling to elongation? Domain 3.2 is nudged out of the RNA exit pore by contact with the growing nascent transcript. Loses its affinity, and now the polymerase loses affinity for the promoter. This is kind of a fun fact page here. The differences between RNA polymerase and DNA polymerase. RNA polymerase can initiate synthesis themselves. They don't need a primer. RNA polymerases can melt duplex DNA. DNA polymerase can't even melt anything open and start. RNA polymerase's initiations prime by a single nucleotide. RNA polymerase initiation is primed by a single non-oligonucleotide. Well, I don't like the way that's worded, because it's primed by a single nucleotide, but it's not really. It is responsible for recruiting that first nucleotide. So I don't know where I got this list, but I wouldn't keep that one there. RNA polymerase make multiple contacts with the 2' hydroxy of the incoming nucleotide triphosphate. Why are they going to be so curious about that 2' hydroxy? They want to make sure there is a 2' hydroxy, right? They don't want no deoxy, right? They want oxy. 2'-oxy. They're making sure, you're a ribonucleotide. That's the one question it asks before it can gain entry. DNA scrunching allows abortive cycling while the enzyme's still in contact with the promoter. For RNA polymerases, the transcript is peeled away from the template, just the opposite for DNA polymerases. In RNA synthesis, many proteins are involved in regulating the initiation, sometimes even the speed and termination, whereas DNA polymerase is only regulated at the initiation step. Get the origin all fired up, then go wide open. Here we see one of the few technique slides, and this technique is now something, this is what the ancient Greeks did. It is so old, and so I'm not sure why we talk about it much anymore, but if you take DNase, of course that digests DNA, and if you just put just a little bit in the reaction tube, you can get it so that it doesn't just chew your DNA down to free nucleotides. You just have it so dilute that it just nicks one strand, or each strand on average once. It's all a probability game. So if you get it dilute so it's not just chewing away, but on average hits randomly your strand, you'll end up with a collection of strands that are different lengths. And if before you added the DNase, if you isolated a discrete fragment with a specific start and stop point, then you'll get, if you ran it on a gel, you'd see all those fragments lined up in electrophoresis, and it'll look like a ladder. That allows you to establish a footprint. What's a footprint? If you do that without any proteins there, you'll get the ladder. But if you add a protein that specifically recognizes DNA at a particular element, as the DNA just randomly cuts, there'll be one region that is never cut because it's got a protein sitting on the DNA protecting it. And so when you look at your ladder, when you run a gel, there'll be one region that doesn't have any bands. It's as if somebody stepped in the middle of your gel and left a footprint. Where there's no bands, that means this area was protected by something in your reaction. And since you added a protein, it's protected by the specific binding of your protein right here and not some other place. That's a DNase 1 footprint. Of course, that requires that you label the DNA only at one end, otherwise you wouldn't see this nice ladder. You'd see just a confused mess. So you have to make sure that you label only one end. How would you do that? You'd use Clinal, wouldn't you? And then you'd have a recessed end, so forth. Contacts one face of the DNA. The book makes a lot about this, but it makes sense. Most DNA binding proteins can only see one helical face of DNA at a time. RNA polymerase is no different. Okay, we got some I guess parlor room facts here that you can amaze your cat. Core binds random DNA with a half-life of what would you expect? That's pretty long in it, an hour. If you bound it and took a snapshot, you'd find that half of it was still there after an hour. Half-life of 1 hour. But that's characterized as loose binding because you can get much tighter binding. Addition of sigma results in a 10,000-fold reduction in binding to random DNA, and the half-life now is a little less than 1 second. Really changes the half-life from an hour to less than a second. Allows it to hop. Of course, it binds true promoter DNA when it's still closed 1,000 times stronger than it's binding up here. So that's got a half-life of 60 minutes. This has a half-life even longer. You see, several hours. Several hours. You don't have to actually go into the melting stage because you got plenty of time. Half-life of several hours. Even tighter binding than you have up here with just non-promoter DNA. An hour to several hours. Specificity, of course, that's the ratio of the affinity to promoter DNA compared to just random DNA, is 10 million. That's the differences in binding constants. The rate of synthesis, 1 per second for ribosomal genes, 1 every 30 minutes for the lac or lactose promoter. No, for the lac repressor promoter, for the protein that regulates the lac operon, the repressor. Starts out recognizing about 75 nucleotides. Remember when we started out, we talked about the biophysicists and the highfalutin calculation about diffusion rates and how long it would take to recognize a promoter. They started with the theoretical assumption that the promoter was 75 bases in length, and that's how it gets it because they did a footprint, and you can see that it goes all the way to 20 bases downstream and 55 bases upstream from nucleotide 1 in the initial phases of recognition, and eventually it gets down to about, in an elongation, down to about 35. So it's about half the size of the initial footprint, is about half the size in the elongation mode. What happens to sigma is originally thought that it always left the promoter, but now we know that about 70% remains associated with the core doing elongation. Well, this is kind of misleading. 70% begins to ride and it slowly kind of dribbles off, depending on how long the polycistronic transcript is. What's the, like this one on exams, what's the ratio of sigma to core and how much polymerase is actually elongating? Okay, remember that pie chart? I put that pie chart for a reason. What's the ratio? Well, you got 25%, 25%, 50%. Only 25% here is sigma bound to the enzyme. That's searching for a promoter and melting a promoter, then it leaves. So 1 to 1, 2, 3. 1 to 3 ratio of sigma to the other, to core. How much polymerase is actually elongating? That pie chart, 50% is bound to non-promoter DNA, 25% is in the initiation stage, and 25% is elongating. That pie chart is pretty important. Well, the two major changes in polymerase's transitions from promoter recognition phase to elongation, one release of sigma and smaller footprint. Recovering from a stall, somewhere, this is not flight school. I've been in stalls in the middle of clouds before. I'm not a good pilot, but I have a license to fly in clouds. And everybody wants to fly with me except people who know me. So anyway, that's enough about me. Anyway, a stall's not a good thing, especially when you don't know where the ground is and which way the ground is. Okay, sometimes polymerase will get ahead of itself and it'll end up misaligned with where the end of it is. The 3' end will be sticking out the front of the enzyme. And that's an impossible situation. How would you solve it? Well, you could crawl your way back in your most humble mode. Oh, please, let's get back together again. Get that 3' in where it's supposed to be so you can start adding nucleotides to it. That's one way, but that's so hard to do. So hard to do. What happens is a couple of proteins kind of come up beside it, and they both get on either side, and they kind of strong arm it into doing stuff totally backwards. The polymerase is trying to do the right thing, it's trying to synthesize, but these two guys get on either side and they go, you're going to have to back up. Instead of making RNA, you're going to have to cut RNA. It runs the synthesis reaction backwards. And wherever it is, it just says, okay, cut. And you go, I don't want to, cut. And so it cuts wherever it is, and that little piece that's sticking out floats away, and now it has a fresh cut in that it now can start adding nucleotides to. Those two proteins are important because they're on the exam. The GREA, GREB, the GRE. Is that GRA? What's that thing you have to take? The GRE. Yeah, but you just take A usually, but this is GREA and B. They convince the polymerase that it needs to be an RNase, not a polymerase. So that's how it gets out of a stall. Which eukaryotic factor performs the same bullying function? You wouldn't know unless you read the book. It's transcription factor 2S, I believe. Yep, S. T, F for transcription factor, Roman numeral 2 for it works with RNA polymerase 2. S because that's just its name. Oh, true or false? Sigma always increases it. I don't like always. Always increases affinity to the core for genomic DNA. Increases the core for genomic. Oh, that means promoter and non-promoter. Nope. Got to be false, right? Because if you're non-promoter DNA, it decreases it. But if you are an actual promoter, you'll find the affinity is increased by 1,000-fold. So it does both depending on the sequence of the DNA. Oh, I just hate trick questions. Most heavily transcribed genes in E. coli fire once per second. The most, once a second seems kind of slow, but we all know it's the ribosomal cisterns. True. Okay, what's the time?
3:27.
It's not spring break yet, is it? Let us keep going here. Heck. Yes. You still sit, but your eyes cry out no. Okay. It's not like we haven't said this before. Minus 10 tau, minus 35, whatever that is. I can't even pronounce it. Upstream. Okay, so this is what the strongest promoter on earth looks like. The ribosomal, a bacterial ribosomal cistron. It has an actual UP element right here, up. This is where each of the C-terminal domains of alpha actually bind. You don't see them very much, but it is used quite frequently in the cell, but largely for the ribosomal cisterns. You'll notice there are other sites for transcriptional regulatory proteins to bind. So let's look at the contributors to its activity. Just by having a UP element here, so you can bind the alpha CTDs, you increase the activity of the core promoter by 60-fold. You can increase that another 5-fold by having these FIS sites. The ribosomal, the RRNB operon, which is a ribosomal RNase, has a UP element. This is the thing that fires once a second. That's as fast as you can do it. The sequence structure, and you know about this, if you change the spacing, you mutate it either towards or against the ideal, and you change the activity appropriately. What type of element is found right there? Okay, this is minus 35. What's right next to minus 35? UP. UP because the alpha CTD likes to make contact with sigma itself, domain 4.2, right? So it doesn't do any good to put a UP element, halfway across the room. The polymerase itself, it sort of simultaneously makes contact with these two sites. Melting, of course, at the -10. Where would mutations most affect the rate of formation of the closed promoter complex? I think we've had this slide before. Closed promoter affected here. The transition to melting to open with mutations there would be most effective. Several sigma factors. I think what I'm doing now is making sure I cover all the slides in the book. Here you see from another text the domains of the different sigmas, even from different organisms. Bacillus is here, a phage, and you see the most conserved domain is this domain 2. I think that's a homework or exam question. This is what both makes contact with the core, and that's where the melting of the -10 occurs. Here you see, now hopefully it's becoming a familiar diagram, domain 2.3, the tryptophan and tyrosine. These make contact, these would be in the domain 2.3, they're responsible for the melting. Domain 2.4 recognizes the TA of tata. Okay, here you see this again. Where is -10? It's actually TAT, it's in that second T. That lines up where the dot, that's where -10 actually is. And now we've got something called the extended -10 promoter. A lot of promoters, especially in B subtleus, about half of them, have things that are upstream of even tata. You have these two nucleotides. This can be anything, and a T and a G is part of the consensus, and that involves domain 3. They actually had to go back and rename it 3 because they found that later than they found out about the other kind of interactions. So domain 3, as if we didn't have enough to worry about. 10% of E. coli have that 50% in gram-positive. 1,1's thought to mask. Here you see the folding back of 1,1 on 4.2, and then when it interacts with the core, it opens up, and now it can find a promoter. Okay, sigma 54. We're through with sigma 70. Sigma 54 is a little bit different. I would call it defective, at least I do on the exam. And I ask you, how is it defective? What's wrong with it? What can it do? Well, it's missing one domain, and it can't do something. There, it's two defects. So the first thing, let's talk about what it's missing. It's missing domain 1.1. So what do you expect? How do you expect it to act? It should go out on its own and try to find a promoter, which is what it does. Because it has domain 1, has no domain 1.1, it doesn't mask anything, it just opens up. But that has risk, doesn't it? What if it started melting promoters and all that kind of stuff? So that's its defect. It can't melt. Except unless there's an accessory protein to help it and some energy, ATP. So let's see how this works. It has a different -35 and a -10, you'll see. Under nitrogen, so domain 1.1 is not present. And so the core has to come to it. It just occurred to me, how does that happen? Because the core is stuck at a half-life of 1 hour. It can't go hopping. How does core ever get there? Does the regular sigma, is it displaced by? That's a good point. You'll have to to hijack, sneak up behind one of these real microbiologists in the halls of microcell science and not let them up till they give you the answer. I don't know how that works. I suggest sneaking up on Shanmugam and asking him. He'll tell you anything. He'll be up in no time. But then you better go look it up. Now, he has an office next to mine, so we always jest. No, he's a good guy. Okay. So I don't know how that works. The first time it ever occurred to me that I didn't know something. So that's what it feels like. So what promoter does this act on? In what we see here, it works on the NIF promoter. This is for nitrogen starvation, but in E. coli it's called the NIF promoter. Also in cyanobacterium, it's called the glutamine synthase promoter. What is in common between these two? If you look here more closely, you'll see that there seem to be two starts for transcription. It's like a dual promoter. That is a trick that bacteria use in order to get multiple ways to regulate. You just stack your promoters one behind the other, each controlled by its own sigma. You have one that's controlled by sigma, minus 10, minus 35, sigma 70, and then you have one that is red hot, that kicks on to a very high level, but only under conditions of nitrogen starvation. So why didn't it ever use this one? It's because the promoter that makes sigma 54 is not activated unless you have nitrogen starvation conditions. And that other auxiliary protein, which I haven't told you about yet, is not induced either. So this thing doesn't work, so you get this kind of slow, steady expression here, but under nitrogen starvation, you get this second promoter kicking in. You know, I wonder if this is not the answer to my question. Because if this one's always going, sigma's going to have to leave, and then you're going to have core skipping down here, and if there's a sigma already sitting there, sigma 54, there might be opportunity for a deal. Maybe so, maybe not. I guess Shanmugam lives to see another day. I don't know, but that, maybe. So back to defective, it's unable to melt. And this is the other protein you have to know about. This is the helper protein, NTRC. I don't know what it stands for, probably nitrogen C or something. And it has its own little element that it binds. And it has affinity, it makes contact with sigma 54. And this is a strange experimental observation that's not completely explained. It has to be upstream, the binding site for NTRC, but it doesn't have to be physically on the same piece of DNA. This is an example where you have concatenated plasmid DNA and you still get activation. In addition, it absolutely requires ATP. So ATP, NTRC are required to melt a promoter recognized by sigma 54. Leaving room for a DNA loop. Yeah, it's thought that there's a looping effect, a looping thing that happens. You know, normally they're not on two concatenated plasmids, it's all on the same thing, but the E element doesn't have to be right beside the core promoter. It can be off a ways, and you can see at least 70 base pairs away. In fact, it must be at least 70 base pairs away. You know, I bet I know why that's true. If it was any closer, it'd be sitting into that sigma 70 promoter, And sigma 70 needs to bind there so it can be channeled into interacting with sigma 54. So it doesn't really need to be 70 away, because that's, who knows how much that is away. It just needs to be on the other side of that sigma promoter. This is how, what's the guy's name? Archimedes and all the medieval scholars just learned everything by induction. They never had to do an experiment. So yeah, just prove that. It's kind of bad. Okay. This whole science gig, it's just nothing. Okay, this is an actual picture of the looping. So we know looping can occur in the configuration, so the NTRC can actually, at least in vitro, form a stable interaction with the polymerase. And of course the polymerase here is holoenzyme, it has sigma 54. And this is a footprint, an actual one, just showing where the sigma 54 is binding. That's why we know there are these two promoters, a dual promoter. And the footprinting, you can see the footprint is right in this region. Should be big, heavy bands. You see a little bit here. You certainly don't see any of these bands. Then the bands take up again right up here. This shows that the footprint is dependent on ATP, not the inactive AMP, ATP. So footprint, no footprint. Footprint, no footprint. Of course, the region of the promoter happens to be one that is not forming lots of ladders. That's the caveat with DNase-1 footprinting. In the textbook, it makes a nice uniform ladder. In reality, it has some preference to where it likes to nick, so you won't get a complete uniform ladder. You'll find some places just don't cut very well, and other places do. So this happens to be in a region when there's no footprint. There's not a lot of bands in here. So you're looking at the difference between no footprint, and then you're seeing a reduction here when there's a footprint. I guess we'll let them publish that, but they're going to have to prove it at least one more way, right? This poor guy never graduate. Open promoter complexes are heparin resistant. I didn't see the heparin. Oh, I didn't see, oh, see what they're doing here, yeah. Okay, that's a concept. So remember we said that in the formation, in the binding of the polymerase and of core plus sigma, then closed, and then open, then ternary, that progression of tighter and tighter binding. One step is irreversible. Everything else beyond that step is irreversible. When you melt the promoter open, when sigma does that, it is so tight in binding that it's considered irreversible. So this experiment shows competition. You can put a large carbohydrate, sort of large polymer that's negatively charged. It mimics DNA in a lot of its actions, negatively charged. And if you have a closed promoter, and the polymerase is bound to a closed promoter, it's not melted yet, you can compete the polymerase from the DNA, from the promoter, just by adding a large excess of heparin. But if you've actually transitioned from closed to open promoter, where you've melted it, that interaction is so tight that heparin is unable to compete. So let's look at the footprint in that light. Core plus sigma 54, and then you have this protein, and now it's plus and minus ATP. So you have everything up here, except the heparin gets in the way, so you want to add the core and the 54 and the NTRC before you add the heparin, and I'm assuming that's what they did. And then if you have ATP also present, you get a complex between your DNA, which is radioactively labeled. It just runs very fast in the gel. But if your DNA, which contains the promoter, is bound to your core plus sigma plus NTRC, it's going to be a very slowly migrating complex in the gel. So here you see making that complex between the promoter and sigma 54 and the NTRC is dependent on ATP also being present. Just so the heparin means that this thing is actually in a open promoter complex. I don't know what this is, but I presume it's the order of addition of the heparin. Open promoter complexes are heparin resistant. That would be the way you see a footprint. I'm not going to show you this on a gel, but these are the kind of experiments you can do, taking advantage of the very stable property of a open promoter complex. Is it spring break? What time is it? We got
About 4 minutes.
4 minutes on termination. I won't do that. I should, but I do have some remnant of humanity left. So this concludes this week's lecture, certainly today's, and what lies in store next week is your own business. I hope you have fun and your cat gets to know you again. See you week after next.